Okay, let's start. I will
Sure.
first introduce and then you can take over.
Great. Thanks, Dr. Mo.
So, I would like to welcome all of you back after the lunch and a very good morning to Dr. Shiladitya there.
Thanks, Dr. Amol.
Yes, so this is the third session for the day for us here.
Right.
So, I would like to welcome Dr. Shiladitya Mitra, who is currently a senior postdoctoral researcher in Max Planck Institute of Psychiatry, Germany. So, the topic which he will be discussing with all of us is emerging technologies in preclinical and basic neuroscience scope for engineers. So before going ahead, I would like to introduce him to all of you. So he has done his BTech in biotechnology from VIT in India. He has completed PhD in CSIR Center for Cellular and Molecular Biology in Hyderabad. uh, his area in PhD is neuroscience, by uncovering the mechanisms of action of an understudied gene called WDR13 in brain. One of the important findings of his research was the importance of this gene in homeostasis and in regulating depression like symptoms. Postdoctoral research at Brain City EMBL. So earlier he was in Nensky Institute of Experimental Biology, and he also worked in collaboration with physicists, optical engineers, and people from the background of informatics. So with this short introduction, I would like to welcome Dr. Sheila Aditya for his session here, and over to you, sir.
Great. Thanks a lot, Dr. Amol, for organizing this. And um, yes, so once you will. Right. Great. So I shall just 1 second. Right. So I guess that my screen is being shared right now. Please just let me know if it is.
Uh, yes, it's being
Uh,
shared.
yeah, it's being shared. Yeah.
Yes, now your screen is visible.
Yeah. Okay. Right. Can you see the presentation?
Yes, yes, please go ahead.
Great. Great. Okay. So
Maybe you
good.
can click that hide button.
Yeah. Yeah. Okay. You're seeing that also.
Yeah.
Right. Okay. I'm sorry. Yeah, sorry. It's somehow there is a glitch in Mac, so it's like
Acha, acha. No issues,
it's
no issues.
I'm just whenever I'm clicking, it's just going away from that.
No
When I try
problem.
to move it down, yeah, exactly. This is what happening. Okay, this should be fine, right?
Yes,
Is
yes.
is down from the presentation screen. Okay. So good afternoon to everyone. What I'll be talking today is basically giving a brief overview a few of the interesting technological areas in preclinical and basic neuroscience. Like I have been working on basic neuroscience for more than a decade right now. And when I started as a molecular biologist, though, like I had a BTech in biotechnology and had a course with informatics. But the more and more that I am delving deep into neuroscience right now, I could see that the field has progressed deeply into depending on technology and informatics. So, of course, like there are multiple areas of neuroscience where there are a lot of importance of machine learning, of neuroinformatics. But I'll be just focusing on two key broad areas which are of interest right now to people, and they will form the basic of neuroscience research for at least the next 6, 7 years to come, and holds immense opportunity for other people coming in from varied backgrounds to put their contribution in neuroscience. So before I go in much into, I just wanted to talk a little bit of biology over here, and a little bit of the background. So brain is a complex organ. It has got different number of cell types, but the most important cell type that the brain has is the neurons. Uh, Everyone knows it is high school textbook. What we have over here in this figure is the action potential of a neuron, because neurons, they fire, and depending upon the exchange of ions between inside to the inside of the neuron and its external environment, it creates a change in the potential that causes uh, an action potential over here, which can be detected by electrodes, because it's conduct through electrical communicating through electric potentials. So what you see over here is a depolarizing wave, and then goes back for repolarization when the ions come back, flux in, and then it goes hyperpolarization and resting phase. So when you see the neurons firing, this is called a firing of a neuron, you'll see a curve like this. You'll see an action potential like this. There have been many, because neurons are electrical in nature, so people have been trying to recreate the function of a neuron. And this is over here is a 3 transistor model of a fast spiking neuron in the brain. These were initially the areas, and these are basically the component, these kinds of designs and of an electrical neuron are basically what's behind neurocomputing. So these were the areas where technology was, during the 90s and early 80s, even technology was pouring in to neuroscience, is to design and have like a model of a neuronal connection. So if you have two brain regions connected by different kinds of neurons, because there are different neuronal types in the brain, so you would try to generate an electrical model of that and see, like, and run it through your simulation to see how they would be interacting and how they would be behaving in, like in times when not many animal researches could be done, or they were severely limited. But as you know that neurons are aplenty in the brain. There are billions of neurons. As you can see over here, this is an image taken from a mice that has been injected with a virus that expresses different like one kind of single fluorescence protein, but then they are truncated to give different colors. And as you can see, this area is completely messy with so many projections of the neurons coming out. This is the hippocampus region of the brain, which is important for memory and learning. So under so you so it's quite complex, as you can see over here. And it's not just understanding single neurons and their action potentials and their electrophysiological properties, but to understand this neuronal ensemble and how they react to the environment is something that we are trying to understand. And the more that we'll understand using preclinical models like mice and rats and monkeys, is that we can translate it to human beings. And like, we can know about the diseases, we can know about how brain functions under different conditions like stress, or when you're learning, or when you're going outside facing heat, you're drinking a coffee, you're having a chocolate, how the brain is functioning, you're addicted to something, you like something. These are all the questions that are yet unanswered. Memory, how it is formed, and through basic and preclinical neuroscience, we are trying to address those questions. So as I've been telling, like, technology, like, all in neuroscience already exists. There has been machine learning, and in cases of fMRI, MRI, in brain machine interfaces, in psychiatric research, informatics is already there in genetic research, where people understand how different gene expressions happening in different regions of the brain. And it's a huge amount of data, and when you have a huge amount of data, you shall require people to do analysis. The most common applications are electrophysiology, in which, as in the previous slide, I told that the action potential. So electrophysiology sees the way these action potential in the neurons are affected under different regions, and you basically do it with the clamping electrodes, or it's called a patch clamp technique, in which you put electrodes and you measure the changes. And it will tell you quite clearly about if there are differences between when you have different conditions. So, but what I'll be talking about is something a little bit different from these tech, because they are already there, and I guess that a lot of speakers who have been there before, because I was seeing the program, it's quite intense, have been talking about different aspects of such analysis. So I'll be skipping those parts because those are pretty common. People know about it. What I'll be going is right now going into more interesting topics. topics which are prevalent. So I'll divide my talk into two parts. First is the application of electronics, where I'll be talking about few where there are direct application of electrical components in without which we cannot do the behavioral analysis or any experiments. And the second shall be machine learning. So I'll go on to my first part, which is on the application of the electronic components directly. And the first thing that comes to mind and the most interesting research right now is optogenetics. So what's optogenetics? If you break down the term, so it's basically optics and genetics. So everyone knows right now that each of the cells of the body, they express different genes. Neurons are no different. Neurons express different genes, and as many people do research, I have done research. Each of this expression of the genes changes or the function of a neuron, how the neurons function. Like I had been working on this gene called WDR13, as Amol told earlier, and then I had been doing an MMP9, FKBP, and each of these genes have a different role in stress, in depression, in the neuronal functions. So what you basically do is that you exploit a principle of neuron that I told, that the neurons exchange ions between its external and internal environment. So what you can do is that if you can express in the neuron a certain kind of ion channel called channel rhodopsin forcefully, and this is usually done with a virus. Yes, the same kind of virus that is causing COVID-19, not the same, but the principle is the same. It causes infection to the cell. It expresses a few of these proteins which are engineered because it's an engineered virus. And once you express this protein, or this is actually a transmembrane channel, what it does is that it causes an influx of sodium ions that opens up the neuron or causes the neurons to fire. And so this is what you do is that, but these channels are actually responsible to blue light. So what you do is that once these channels express, and if you shine a blue laser onto the neuron, these channels will open and let the sodium in, just causing a firing of a neuron. Or what you do is that you create an on switch for this neuron. And the similar thing can be repeated differently by using another kind of protein called a halorhodopsin, which on emission of yellow light causes influx of chloride ions inside the cell, just changing the potential or shutting down a neuron. So this is called an off switch. So basically it is an on and off switch that you can use to turn on a neuron and turn off a neuron just by expressing these kinds of proteins and by light. So this is called optogenetics. This has an immense potential these days in understanding how neuronal ensembles function, because you can express them in different regions of the brain and shut or on or shut off its function, and thus you will be knowing that which small neuronal subtypes or which brain regions has different function. As you can see over here in this image, it's a mice which has an optical fiber inserted
Dr.
into its.
Shiladitya,
Yes.
sorry to interrupt.
Yeah.
Actually, some voice is coming.
Yeah. Yeah.
Uh,
Actually,
yeah,
it's not mine.
let me
Yeah. Sure.
identify.
Sure. Sure.
Uh, Vimlesh, are you there? Can you please? Vimlesh, actually, I am unable to remove also.
Yeah. Yeah. It's.
Vimlesh, okay, I will try to get mobile number and try to contact.
Okay. No worries.
Yeah, please go ahead.
No worries. No worries. I can hold on.
Yeah.
Voice.
Okay. Muted.
I have
Yeah,
muted.
correct. Please continue.
Yeah.
Right. Okay. So what you can see over here in this image of this mice is that an optical fiber has been inserted into the skull of the mice, and the blue light is being shining, obviously going to have some effect on this behavior of this mice. expressing these genes. So these, like coming from most of you guys will be from electronics, electrical, informatics background. So this is an active area of research, and there is a lot to be done over here, and particularly if you're coming from an electronics background. Going to the an experimental paradigm, I'll just tell about an experiment that I had performed in brief over here. So as I told that, like there can be on switch and off switch. So there are both experiments over here done. So there's a region of the brain called the amygdala. So what I did was I expressed this virus, which was expressing channel rhodopsin, which is the on switch in the but don't bother about this now. What this figure tells over here is that when you give sugar, which elicits a liking kind of response, because everyone loves sugar. Mice loves sugar particularly, and you can see as compared to the control in the experimental animal, which has been given sugar an appetitive preference task, like it's called technical jargon. You can see more neurons in this region, which is the same region, getting fired up in a more synchronous way. So that we know that, like this region is involved in preference, and like and mice, when it likes sugar, like these neurons, they glow up, they turn up, they show more activity, because this is a CFOS labeling is a marker for neuronal activity, or actually plasticity. So then we decided to, like what will happen if we shut these neurons off over here, like will it affect the function of the mice or the rat? So we did this experiment in rats, and this is called a Skinner box experiment, in which I'll go shortly through the behavioral paradigm. Is the mice goes and presses this lever, and it gets a food dispenser over here, but it is trained through loudspeaker lights and electrified grid to, like go and press the lever on its own. And so technically the idea is that whenever the laser, whenever the loudspeaker and the lights are on, the mice gets a response to go over there, and it gets stopped, it freezes, but if not, the mice goes and gets the food from there. We express in this sorry, not mice, rats, this halorhodopsin, which in this amygdala region, which we saw earlier, was involved in place preference, or this liking thing towards sugar. And we, of course, had a laser as similar in the previous picture, lighting onto this region as through the optical fiber. So what this graph shows over here is that normally when you have the sound on and the laser on, and without any of this affecting, the rat can go over there and get the dispenser from the food. But when you have just a laser on and there is no training over here, it shuts down the neurons, and thus shuts down the likeness that this rat will go and press the lever and get the food. So it reduces the appetitive, or this having sweet water preference, as compared to the controls, or which are the mice that were not injected, or injected with some virus that is not expressing this, and increases the freezing behavior in the Skinner box. So this is like a kind of experiment that many people are doing right now, in which like they are trying to understand the neuronal ensemble function. And as I told you, that because of the component of this amazing concept of having this optical fiber emitting light onto this ensemble of neurons, you can control the behavior of these. In a similar manner, what we have, because I was telling, because about electrophysiology, in which the first slide I showed that you can check the axon potential. Those were done mostly from patch clamped, or cells which are grown on cell culture, like neurons which are grown on cell culture. But when you'll be doing in vivo, like a mice over here, like which is freely moving, so what you design are something which are called electrodes, multiple electrodes, or even better are optrodes. So optrodes are derived from multiple electrodes, which you'll be putting inside the brain. And when you put multiple electrodes inside the brain, it will give you a reading from different layers of the brain, because each of these small dots that you're seeing are basically electrodes, and these are the big spikes. So you'll be getting information from the neuronal activity from different layers of the brain through each of these electrodes, and thus generate a huge amount of data. But what if we wanted to tie it up with the optogenetics principle that I told before? Like you shine light, you activate one region, or like you shine light and deactivate one region, and see how the neurons are behaving over there, get the information, electrical component information out of those neurons. So design something called an optrode, which has the optical fiber and those electrodes over there. So it looks kind something like this, in which like the blue laser shall be emitting a light onto that part of the mice brain in which you had injected the virus, thus causing changes in the neurons over there, and then you record these with the electrodes, the signals and how the neurons are firing. The kind of information that you get over here is something like this after you run it through MATLAB or Python programming, because first you'll get raw data and then you'll get a heat plot like this. And there are several lines over here. Actually looks like a painting, but there are actually several lines over there, and each of these lines represent one single neuron, and the firing of each of these neurons. So you see over there that like in general, apart from this small block over there, so this is the general firing of the neurons that you will be expecting when like in that particular region from where the analysis is happening. It's more or less uniform. But when you have this blue light shining on, and this is basically or like causes more firing of the neurons, you see that there is a change in the activity that you are recording from here, particularly in this small time interval during which the blue light was on. So this is how you measure the behavior of the mice. Because the mice is free moving, so it can do its behavior, though there is of course something attached to his head, but after some time mice gets used to this. And you can know about the behavior of the mice. You can record a lot of data. And I'm sorry, am I audible?
Yeah, yes, sir. Please go ahead.
Right. Sure. So yes, so as you can see that you can get a treasure trove of information from this, but of course you require a lot of electrical components. As you can see, people of you who are like involved in electronics again will be able to appreciate it more, and also for people from the informatics background. We are end users, so we have the MATLAB libraries plotted out so that we can get, when you churn in the data, we get data out like this, more or less standardized of how to get this information out and reflect the behavior of mice. In similar manner, there is something called fiber photometry, and what I can do is that actually I can show you a video. But let me tell you first is that as one principle that I told is the action potential of a neuron. There's also something called the calcium wave of a neuron, in which the neuron takes in calcium and then it glows. So if I can go back, because I had kept a YouTube video. So if you see over here, these are some neurons which are injected with a dye which emits light when light shines on it, so that we can see the neurons inside the brain, because otherwise you cannot see anything. And then you'll be seeing what is this visible, this YouTube video?
Yes,
Yes,
yes.
Right, okay. So you can see over here, if you can see, is that yes, you can see those neurons glowing. So this is what is happening, is that there is a calcium flux which is changing inside those neurons, and thus like you can see this beautiful glowing of the neurons, the neurons throbbing actually. So I didn't have a video of mine, so I had to take up a video from the internet, but I hope that solves the purpose of it. So anyway, so again over here, what it does is that this is a fiber photometer, and this is called a fiber photometry, in which you put an optical fiber again into, because those neurons won't be visible until unless you show the light on that, and then this optical fiber also records information back from the region of the brain where the virus expressing, in this case, the GCAM6. And this is different from the electrical component, because this shows neurons which are active and the population of neurons which are active. So suppose you are interacting with someone or interacting with something. So there are certain populations of the neurons in the brain that will fire. So this is one way that you can see which neurons firing, because there are multiple neurons in the region. So it's not that all the neurons will be firing, but a subset of the neurons will be firing. So you can see the subset of the neurons that will be firing using this technology. And again, this would not have been possible if there would not have been a development of the electrical or electronics component of this, which is exemplified in this figure, in which you have a bidirectional optical fiber which excites the cells as well as takes information from those cells, and then transfer this information back to the computer into a more usable form. Then again, you run it through my Python or MATLAB, and you get data out of it that's saying that how many neurons and what's the spatial distribution and what's the sequence of firing of, or what regions they are firing. So a lot of information you can get out of it. What's shown in this figure, like how it looks like, it's around 5mm, the longest ones 3.5mm, and each of them has got multiple like the ends has so you can a mice, this will cover multiple regions of a mouse brain, because a mouse brain is small, and get a treasure trove of information out of it, like how different regions are firing. So I told you like 3 examples right now of and these 3 are particularly used immensely in understanding the behavior and the firing of neurons. And if you are coming from electronics background, there is a monopoly actually of one company called Thor Labs, which designs these kinds of components. And if we have to order or purchase any components, we usually just go to Thor Labs because they have the premium service. The thing is that because they have almost a monopoly in the market, the prices are quite expensive. And I would love to see, some of you would be, turning entrepreneurs, or, or many other entrepreneurs will be coming up to design electronics chips, which are important for, or design some changes in some electronic boards, which will be enabling us to do such experiments as exemplified before, or, design some more different kinds of experiments. So if you are interested into, that, electronic and electronics components are used in basic research of animal research, you can just go to Thor Labs, you can just see what they are doing, what are the different components that they are making, and how you can be implementing those into your research, or into your entrepreneurship, if any of you would be interested in that. So this was more or less the electronic components in of the different electrical aspects of basic neuroscience, which are super interesting. I'll go back right now to something which is related to machine learning in neuroscience, in basic neuroscience. So when I talk about machine learning, of course, like you have heard, you guys are from informatics or engineering backgrounds, so this is something new to you. You have been hearing machine learning in fMRI, in MRI, in brain scanning images, in different applications, in biomedical engineering, different applications. So you know about TensorFlow. This is the most commonly used right now for neural networks and for generating algorithms. And the few softwares that I'll be telling henceforth all use TensorFlow for their deep learning and for the neural networks and for the training purposes. It's pretty easy, TensorFlow, to integrate into your software because of obvious reasons, which you guys know. But then just for the sake of it, what it does over here is basically, as in school, you would be doing some empirical formulas, like you are given like a set of data and then you generate a formula to fit it. So
Hello,
this is
sir.
what it, yes?
Yeah, sir, sorry to interrupt you. I just want to inform you that few participants are from pharmaceutical background and a few are from biomedical as well.
Okay, so then I'll just explain a little bit in details.
Yeah, thank you,
Uh,
sir.
right, sure, of course, great. So then TensorFlow, what it does is that you can get it easily. It's actually supported by Google right now. So anything that you see in your smartphones, you use Google Lens. You look at something and you see that what object it is, or you scan a QR code or any of these stuffs. They're all based on deep learning, machine learning, or you are taking a selfie, you just smile towards the camera clicks. So they're all based on algorithms of face detection that Facebook uses. They're all based on deep learning networks. And TensorFlow, if you are a beginner or even if you're an advanced guy, TensorFlow is the most easiest of them all. It has got very simple algorithms. You can incorporate them. You can learn it. You can just design an app to do that. And how TensorFlow works, this is a very simple example of data fitting, is that if you have a set of data for X and a set of data for Y, and you do a model fit that X is equal to Y, and like it just fits one point to another point, and then you run it through 500 times iterations. So what it does is 500 times it will be like fitting the different iterations, running it through the program, similar to that kind of that you have been doing in school days, that you have been given a set of data and you generate the closest equation that you can get, like a is equal to b, or a plus b is equal to c, or if you have a corresponding a, then what will be a corresponding b. So this is what the machine does over here. Like it does not have equation. What it does is that depending upon what you have fed into it, so it tries to keep on corresponding to it. And the more number of iterations that you give, here I had given like a 500 iterations, you can go 1,000, 10,000. So the machine runs it, and the more times of iteration it does, the lesser the loss is, and the lesser there are the chances of error. So when you do, like you say, like a print model predict 7, in which you have a given input over here, in which -1 would be corresponding to 3, 0 to 4, 1 to 5. So you will be getting a value which is close to 11, but may not be completely 11. So this forms the basic of a neural network and machine learning, and this is what is being used in all the future subsets of the programs that I'll be talking about. But this is the basic of it. Like you generate a training network based on your model, and then you keep on training it multiple times till the point of time your error becomes less, and it comes close to the actual value that you are supposed to get. But instead of doing it by yourself, when you have a large data set, you just have a network to do it for you. So one of the first applications for this is brain functional and projection maps. So many of you who are, again, like from biomedical and pharmaceutical backgrounds, you must be seeing the projection functional maps for in case of fMRI or MRI, and you'll be seeing how, like if there are activity changes or if there are changes, they're plotted onto certain regions of the brain. And you just go and you say, when you finally get the data, you say, oh, this is the region of the brain which is getting affected. You have changes of activities, say suppose in the hippocampus, or say, suppose in the amygdala, or different regions of the brain. But when you're coming for analysis in cases of mice brain, like this is getting more and more interesting, because we are delving deeper. We are deeping into single neuron functions, single neuron functional and projection maps, which will help us to understand and extrapolate it to human beings to understand a greater picture. So again, I'll give a little bit of biology. So what's shown over here in this picture is the advancement of a certain kind of technique which has been developed over the past 5-6 years. Actually, it's a very recent technique which is called clearing the brain. So this is done post hoc, or after the experiments performed. So what it does is basically any tissue that you will get something like this, like your muscles, the brain, because it has lipids inside it, it has got pigments inside it. So, like it will be opaque in general. But what this technique does, there are like 4 techniques described over here, the 4 most important techniques, is that they optically clear the brain. So what they do is that basically most of them take out the lipid components and retain the structure of the brain, so that the brain structure is there, as well as the internal components. So that's the reason you can see these beautiful neurons when you do microscopy on these brains. And there's a special kind of microscope that is used to analyze these brains, because they're not just slices, they're not just one section, but it's a complete 3D brain. And it's called a light sheet microscopy, because you have basically the scanning is in one plane or one sheet of area. So, or it's a light planar microscopy. So you get information of the neurons in the different regions of the brain, and the entire map over here of the brain. Thanks to the clearing, like a normal microscope is limited to how much it can penetrate inside the tissue because it's opaque. But because of clearing, you can go much into understand the finer structures. And if you are from someone from the pharmaceutical background, I know a couple of doctors who had been working with us when I was in my previous lab, who were analyzing different molecules in different regions of the body, not just brain, because I'll be just talking about brain, but they were analyzing the heart, the muscles, and employing these techniques to understand the distribution, the kinetics of these under different experimental conditions. And I'll show a short video over here because I stained the brain against one molecule called DARP32. And when you can see over here, this is a clear brain. That's why you cannot see much of the outline. But what you can appreciate are these regions which are like highly stained, some sparse staining, and there's beautiful projections that are coming from one region to the other region of the brain. So this is what we will be seeing more, and you and people in neuroscience are doing more, because the time has gone when people will be focusing on one region, and right now people are coming of this advance of technologies is focusing on the entire brain regions and the distribution of different molecules, seeing the different projections. But you have a treasure trove of information over here. If I would want to understand that which region in like my region, how the intensity is, then what I have to do in normal conditions is have to open a reference map of the brain. Here, this is a mice brain reference map. This is the region which is called the amygdala, which is involved in fear learning. I'm sorry. Yeah, right. I thought, like I was not audible. Am I audible, right?
Yes, sir.
Yeah,
Yes,
okay,
sir.
yeah, okay, great. So what you can see over here is this is the amygdala, which is involved in fear learning and memory. And this is the same staining as earlier. So if I would be doing the analysis, I would be, so suppose so this is an experiment in which I stressed and postnatally and having seen the changes in the brain. Then I'll be manually pulling this reference map up, compare, because it's light sheets, so I'll be sectioning it in one sheet, and then like seeing each section by sections, what are the differences. It's okay if you have like a region of interest, like in this case I had a region of interest of the amygdala, so I can see in this region how the changes are there. But if you want to see throughout the brain, it gets next to impossible. So in this case, like when I was showing you the firing of the neurons in um, the changes of the calcium levels, there is another way that you can check for the, let's say, plastic or activity of changes of the neurons, and it's called using a marker which is called CFOS, which is an immediate early gene, but doesn't matter. Over here, there's a brain which has been stained with CFOS. It marks and labels most of the neurons that has been undergoing changes or undergoing activity changes, and this is how it looks like when you see the brain in details. Like it's completely packed. There are a lot of these cells that has been labeled, and if you would want to get information out of this in general, it's next to impossible if you would want to understand and compare it between your experimental and between your controls. So here comes the usefulness of a software which was developed based on Python and TensorFlow, which is called the ClearMap. What ClearMap does is that it has basically two important components within it. One is called a wobbly stitcher and one is cell map. So the wobbly stitcher has the same algorithm as you do like when you're shooting a panorama, like like it overlaps the images and then it creates a more fluidic picture instead of having these gray zones which you would be having earlier times in the earlier panorama, which would be taking each frame by each frame and then try to stitch those frames. But instead of doing stitching of the frames, what you do is that you overlap the frames and take up the most common regions, and then you have a more fluidic thing. So that's what exactly this does. It does a wobbly stitching job because you have to compare it to the reference brain that I showed over here. So the idea is to have the brain stitched up completely and then superimpose onto this reference brain. So that it can be more fluidic and there are no gaps, because then you'll not be understanding which region is which and where it is more expressed. And then you have the cell map. So here what you do is that you train the network, and you train the network to distinguish between a single signal and what is a background and what are cells, and then you just put it into it and it derives and generates a map. So how does it work normally is that you correct the illumination because it's a whole brain, so different region will be illuminated differently. You do a background removal like it has been done over here. You do an equalizing and the Gaussian filter, the maximum detection, cell shape detection, and then cell intensity. So what you can, okay, I'm getting an error message is, I'm sorry, just. I'm sorry, I'll just restart it once. I'm sorry for this. Hello, hello.
Yes, sir. Yes.
I'm sorry. I think I just cut into. Okay. I shared the screen. Is it visible? The screen?
Yes,
Yes,
screen is
sir.
visible.
Yes, yes.
It is visible.
Right. Okay. Because I'm not getting that message anymore. Okay. Anyways, right. So I was going back, like how you train the network and how you. So what you do over here is that now you select the regions which you mark it as cells, and then you label them, and then you run, like you capture each frame by frame, because over here, as I showed, that like there will be, so this, if this is the entire plot, if this is the entire plot, you will get each frame by frame something like this. So then you go through different frames, you label, and then you run through a network. And when you run through a network, then you have your network trained, and then you just apply, and then you'll get the data. So this is one example of a project, which is not my paper, it's someone else's, but this is a more detailed one, so I thought that this would be easier for you to understand. So what they had done over here in this experimentally is that they wanted to know which neurons are heat sensitive. So if you are given, suppose if the mice is subjected to 38 degrees Celsius, which of the neurons they fire up. So this is the control brain, and this is how it looks like. They have done the same CFOS staining of it, and using multiple brains, and then superimposing and generating a p-value map. And these are the heat maps. You see that the neurons which will be firing, and this is all generated using the clear map technology that I just talked about before. And then when you have the mice which has been exposed to more heat, then you have different regions of the brain firing up, and this is what is exemplified in this figures over here. And when you superimpose onto the atlas, which is exactly what ClearMap does, then you can generate a map plot like this, which are the regions of the brain where you get activity under different conditions, which has a increase in activity, which has a decrease in activity, and these are all can be done very easily. So once you have your network ready, you just put it, and then it just runs this iterations, mapping it onto the cleared brain onto the reference brain, and then plotting the different cells which are expressing CFOS onto the reference brain, and then giving you this analysis output. A similar thing that can be done is that, as you saw in my picture over there, there are a lot of these projections. And if I would be knowing that which region to another region these projections are going, or have like an entire projection map, then manually doing it is impossible. So what you would do is, like, you use another software which is called a trail map, or you can use, if you are programmers, you can just generate your own thing, because it's again based on TensorFlow and Python. And what it does is that over here on my left hand side, this is example, like you have again, because it is each section, so like you have one section, because so what it does is that you take each plane, and it is a 3D, right? So you'll be seeing different small, and then when you stitch it all up, you'll see the bigger picture. So what it does is that you have one image capture over here, and then you do a threshold application, and then you go ahead and label that which neurons are, like which of these are axons, which of these are backgrounds, which of these are projections or not. So it works in the same format. So what it does is that we have a software called ImageJ, which is this, which I had been using to show this example to you. And you load like each frame like this, and then you label each frame as what is the background, what's the axons, what are artifacts, the age of the axons, and then it does in a 64 pixel frame. So like you repeat this multiple times, and then you train the network. And then when you train the network and then you do something called segmentation, then you see that this was the original 3D projection image of a small region, and you would see that it's quite faint, and if you want to gather the information out of this, like which regions, manually it would be quite different. But when you have after the network has been trained and you do a segmentation map, and then you generate the data out of that, then you have something like this image over here, which is basically the same, but you see everything even more better, everything in more clarity. And this is something which is immensely useful in tracing the different regions the axons are going, like if the axon is going from the end of the brain towards the beginning of the brain, or circumventing different regions. So this is immensely helpful, and it can be done easily using machine learning. This is again another example. What you see over here, there's few of these neurons that they have stained, and you can see with the heat plot that like which regions are the neurons, axons, projections are more. And this is a certain kind of neurons which is called the orexin neurons, which is involved for hunger and food. And you see that which regions like these neuronal projections are there, saying that which regions shall be affected by this, which is a very important information, knowing that because if there is in the amygdala, which is involved in fear learning and involved in also in appetitive preference, as I told during the optogenetics experiment, that you have projections over there, meaning that those in areas might be getting affected. So, and you can see beautiful meshwork of the projections, and this data has been taken out from the trail map. But this is a much more refined data of the trail map, and but you can get data like this out using machine learning algorithms. And the last interesting thing that I would be talking about is using in animal behavior. So, like people are from any that in preclinical and clinical experimental research, we use mice and rats to do our analysis of experimental paradigms. We run it through different behaviors, and then we do behavioral analysis. So, for example, this is one of my old experiments in which, like I had put a mice. So this is how it normally works, is that you have a software, you put the mice, there is a video tracker over there, and the mice does its movements, and then it track and generates a data. So in this experiment, what I had done was you have two objects in a box, and basically you replace one object with another new object, which is here. You replace this object with a new object, and then you see how the mice behaves. When first you make the mice train with having two similar objects, then you change one object with another object. It's a very simple example of which we use to understand the cognitive ability of a mice. And basically, if a mice is cognitively fine, then it should explore the new object more because it has innate sense of curiosity. And this data has been analyzed using a software which is called ATAL, which tracks the movement of these mice. And as you can see over here from the plot of the movement, the control mice, which is marked by wild type over here, has more exploration towards the new object, which you change. But however, my experimental mice did not show this same kind of pattern and tried to stay with the old object more. So these tells, and this tells that in my experimental mice something is different. So this kind of data analysis is fine. Like if we are using a pretty straightforward data analysis of mice movement and behavior, using a software package like Ethovision XT or any maze, there are multiple such software packages, and there are a few open source as well. So like they're still fine. Like there's Bonsai, which is also based on Python and learning. So like it's fine. But then the difference comes when you have to do multiple mice over there. So if you have two mice interacting with each other and two similar coat color mice, both are black or both are white in color, this software can't tell the difference which mice is which. Or like if you have a home cage experiment in which you do not want to disturb the mice from their own home cage, and you want to keep the mice in their home cage and see the exploratory behavior, how they're interacting with each other, which is a more natural way of understanding biology, because biology does not happen in isolation but in interaction. Those things are completely not possible with these proprietary softwares. So here comes the importance of a new kind of software that has been developed, again based on TensorFlow and Python. It's called DeepLabCut. It's an open source, and this is basically the how DeepLabCut works. It's pretty straightforward. I'll go through a few codes next page, and I feel that in the coming next 5 to 10 years, this is going to be really immense because of its application not only in rodents and preclinical model behavioral research, but also in research of any other and human beings, for example. So what it does is that it create a project, you take a video and then grab frames from there, and they have a GUI as well as a terminal based approach, mostly works on Linux but can work on Windows and Mac as well. And then you train your network based on a neural network, which is again based on TensorFlow, to which of these are what the mice is doing, how they're behaving, and then evaluate the network performance and run the videos. And it sounds very easy, but then it's quite tedious, because you have the labeling part and the training part is the most difficult part. Because after that, when you have the results, then plotting the results and getting it is more or less easier. You can have a MATLAB program to get whatever information that you want to get out of that. So it's pretty much fine. So a GUI of it basically looks like this. It's for people who have lesser programming abilities or end users, more like me, because I'm not a master of programming, but I can do tinkering around a little bit. So what it does basically, it does exactly the same as you would do in a command prompt. As you can see that when you run the GUI, then it runs on the top of a command prompt like this. So basically when you click each of these, is basically sending a command, and you can see the command. You can actually drag it down and do some changes if you want to change some of these iterations. So and it has but the good thing of this GUI is that it has exact like a flow chart, like you do it each by each, and so it has a streamlined pipelined output of what is supposed to be done. So what you do basically is that you first create a project. This is basically all the crux of the programming that you'll be using. Of course, you'll be doing some changes in the programmings and like modifying them, but it's more or less a 12-step approach. So what you do first is that you create a project, like the first normal thing that you would do. Then you add the videos. Suppose the mice are moving or the rats are moving. You generate a config file, and this config file will tell you like what you want to label. You want to label the digits of the fingers. You want to label the ear, nose, body. This is what people do if you want to be more precise. And then you do something which is called a skeleton builder, in which is like this, in which once you have labeled it, then you draw and drag a skeleton. And this is very important because once it actually grabs the entire mice over here, and when it will be running through your video or training, then the entire plot is important. So generating a skeleton is an important part, and it can be done by simple drag and drop approach. Like it's pretty easy, because even though this has been this one is the command based one, you can do it pretty easy. And then what you do is that you extract frames, because when you have video, then you extract each frame, like a mice moving a little bit, then a little bit, and then you have to label each of them tediously. And this is something that you have to do manually. But once you have done that, then you just label the frames, and then you create the training data set. Once you have created this training data set based on these labeled frames, then you train the network. That like these are my labeled this is my based on this config file, that these are my config. I have labeled the ears, the nose, different spine regions, the tail, and then you run it and train the network. And when you train the network, you at least have 15,000 to 30,000 iterations in which it will go on repeating it, so that as I told in the first, the error chances will get lesser and lesser. So you can see so basically you are making the machine learn. and then you do an evaluate network. So what evaluate network does is that it gives back another video to show you, and then you can, like, check if everything is fine, or like, or then retrain the network if you think that something is going out of tune, like it's not going correctly. And then once you have trained the network based on a couple of videos, then what you do is that you put it on a batch of videos, because when we do experiment with mice, you usually have experiment with around 40, 60, 70 mice, and then if you have to do analyzing by yourself, it's extremely tedious, particularly if you're doing multiple behavior analysis. So then you do an analyzed video based on this neural network and the training network that you have generated. And once you have the videos done, then you have to get the information out. So you convert this into trackless. I'll show you a picture soon. And once you have the tracks of the mice, then you just convert it to H5 and you get the data out. So it's at the end, it looks something like this. So when you have, like, this is basically the labeling approach. When you have, like, and if you are in a GUI, you'll be labeling it like this. If you're not, then like you'll be labeling it to the command prompt. So but then anyway, this window anyways opens, not this if you are using the command prompt, but this labeling window always opens. So you label the mice, and what it does over here, the beauty of this software is that you can have 3 different mice of the same coat color, and it can then distinguish, because you can have different markers. This is yellow, this is orange, one is pink, and they will keep on tracking these mice throughout the video as separate entities. And when you have that, then you, at the final end, when you have run it through Deep Lab Cat, you have something like this, like, this is mice 1, this is mice 2, this is mice 3, how they're interacting with each other. When you see the video, then you'll see like these points that you have plotted move around, and then you can extract the frames, and then you can extract the plot out of it, like the track plots, and then you can understand, how these mice are interacting with each other, and whatever information that you want to get out of this. This is pretty straightforward. However, as I told, that training the network is immensely complicated, because you have to do, particularly if you're doing with multiple mice, you have to do it multiple iterations and retrain and train and train and train. That requires hours of work. But once you have done it, the analysis is pretty great. And this has immense application, not only just in behavioral neuroscience, but people who are in experimental research in wild animals, or in other, or human behavior. Like, I remember the times when someone complained that Muthaya Muralidharan doesn't ball the ball like properly. He uses, and people were using different trackers to check the way that he is doing, to check his tracking movement. But this is something which is label-free tracking, because what you are generating are virtual labels, and you can generate a complete data out of that. So if any one of you are interested in such kind of analysis and not just animal behavior, you can use because it's an open source, you can just use this software and like tinker around with it, so that you can develop your own program. An example over here is this racehorse, that is shown in Deep Lab Cat's file, in which you train the Deep Lab Cat to note that different regions of it, and when it is walking, so that like the background, and you train the network based on this. And then you can have a video of it, and then it tracks it throughout the video, and you can use it for checking, like in this case, this was done by a person who was into horse breeding, and thus he wanted to see the performance of his horse, and it can be done in race tracking, it can be done. So the applications are quite immense of this kinds of machine learning approaches in animal behavior and in research. So I guess, like, this gives a more or less a crux and an information about what are the different areas that like are happening right now in neuroscience. Like, you have different electrical components, people who are interested, and you don't have to, when you think biomedical, you think only of human applications, but you have an immense scope. And if you're an entrepreneur, particularly, you have a great deal of advantage, because the market is still free. And if you are a programmer, because I know a few people who just did their BTech in informatics, and then they came down to work for behavior analysis, and it's a full-time job to sit behind and do an informatic analysis of the behaviors or generate the data, and particularly in pharmaceutical companies, you'll require it more and more often to implement these data if you're working in animal behavior research or something. This thing's something that you can see to be used everywhere. So in overall, what like it can be said is that experimental basic neuroscience, you would see some old papers from the 90s, would be mostly based on molecular biology or a little bit of electrophysiology or something, or but not much of technological aspects. But more and more that we are going 20 years down the line, 2020, 30 years down the line, over here we are seeing an immense growth in the application of technology. And the examples that I told you before, like without that, right now, no behavior or no biology is possible. So there is an immense scope of interdisciplinary research in basic neuroscience, even not just in clinical neuroscience. And I hope that I had given my point across. And if you have any questions, like please just shoot. Hello.
Hello.
Yes.
Yes, sir. Let me check. Actually,
Yeah.
so open to questions.
Yeah, I'm open to questions.
Okay. Okay, means I had one question,
Sure, go ahead, Dr. Amol.
like as a part of curiosity.
Mm-hmm, mm-hmm,
How does this optogenetics or fiber photometry works? Is it
mm-hmm.
similar to photoplethysmography?
Uh, well,
Like,
I am, yeah, yeah, you're saying something.
because
Please
it
say.
is also based on the principle of light, means blood flow through the vessels,
Yeah,
something like that.
yeah, exactly. Over there, you'll be detecting like any of these techniques, like PET scan or what the one that you are talking about. So they do the same, but here, what it does is that you are directly affecting the function of a neuron based on light, because you are expressing a protein in the neurons extraneously. So this changes the physiology of the neurons. And when you shine light, it just causes the neurons to fire or stop firing. So this is what how optogenetics does. And what you do in, but the analysis component, like when I told about the optos, is almost the same, because you do the same with blood flow analysis, I guess, by checking the light through and how they are changing, how the phenotype is changing, the same. But then the thing is that over there you'll be checking mostly non-engineered, let's say like this, non-engineered cells or non-engineered tissue. But here they have been engineered. So if you have not engineered them, if you're not expressed specifically this protein called channel rhodopsin in these neurons, they will not do any effect. So just shining light on them will not have any effect. But only because this protein is expressing that you're changing the physiology of there. And that's what you are right now analyzing.
Okay, okay. So one more is there, sir. How neuromorphic computation and
Mhm.
neuroscience is related? Neuromorphic computation and neuroscience.
Neuromorphic computation. Okay, wait. I'll switch off from there. Yeah. So in case of neuromorphic computation or particularly neuroscience, so when you see of neuromorphic computing, basically what I told is that you remember my first slide in which I was talking about this three transistor model, right, of the neuron. Uh, Wait, maybe I can just wait. Maybe I can just go back to present and uh, yeah, okay. So let's go back to the first slide over here. Yeah. So when you see over here is that this is the example of a neuron in which it has been designed through a three transistor model. So what is corresponding over here is a fast spiking neuron, which are typical of GABAergic neurons in the brain, which are there. So when you have a model of this and you have different such models, then you generate a model of a neuron. And this is what people had been trying to do, is that mimic the function of the brain. This is what I said in the beginning, like this were the crux of neurocomputing and earlier, and like and even right now, in which what people try to design are artificial neurons based on these same principle. And then when you give a stimulus, and then it works out. So this is also a very active area of research, but it has been there for quite some time right now. And people have been trying to develop artificial brains or artificial systems based on this. So yeah, like that's an important part of neuroscience. But but that's a more important part of an application based neuroscience. And like if you're more of into electronic components or something, this is what you'll be mostly interested in. Or brain machine interface for that sense, like that's again based on a circuit diagram, which is called a circuit diagram like this. But when you come to clinical neuroscience, you have to first understand how the neurons are connected to each other to generate a circuit map like this. So at the end of the day, we are all working towards a common goal to understand how to generate the brain map or a complex map like of the brain function, and these are all aiding towards that. So generating an artificial brain, yes, that might be the final goal. And these small techniques that I told are aiding towards understanding that, because you need to put each of these informations in to when you're generating a proper brain. And I feel that with the advancement of technology, it can be more easier, because you'll not have just the input and the output, but also you have to couple it to the behavioral changes and how the different networks interacting with each other. So yeah, if I've answered the question.
Yes, Professor Vivek, would like to go for extension of your question? Okay. So maybe means if you can extend, like what whole new world of opportunities are opened up through this optogenetics or fiber photometry?
What we can see right now is that it is right now used in every lab. In India, particularly, I'm yet to see, mostly because of the. But it's not that we don't have money; we have a lot of money. But still, it is to be implemented somehow. At least when I was leaving the country 4 years back, this technique was not established anywhere. But when you come abroad, it's used everywhere right now. It tells, because people have this is something that I feel that it's going to have immense use, particularly optogenetics. It's still a long shot, but it can be used in treating patients, even with psychiatric disorders, or making a brain machine interface later on of people who have neurological issues. But that's still a long shot. It's still an experimental design right now, and that's exactly the reason we need to refine it. So what fiber photometry, optogenetics, and optos they do all together are basically refining the same techniques. Because in optogenetics, what you do is that you express this virus, you express into this entire region, entire population of neurons. But you don't want to do that. You want to express in a subpopulation of neurons. And this is what exactly fiber photometry does. Fiber photometry gets the information of which subpopulation of neurons are actually firing. So you do not express in all the neurons, but these population of neurons. So suppose you see an image, and then this image creates a memory map in your head. And these are based on certain neurons firing in a certain region of the brain. Suppose hippocampus, only certain neurons fire, not all of them. So manipulating all of them genetically is not something that you want. You just want to manipulate only those small. So what we see in future is that these kinds of selective manipulations that will enable us to recreate the memory, implant memory, like all sci-fi things. But this is what people have been doing right now, and a lot of this is not science fiction, but is proper science. People have been trying to create artificial memory. People have been trying to create trace back the same exact memory by seeing the sequence of the information flowing through the brain using this fiber photometry and optogenetics.
Great, anyone else?
Sorry, go ahead. Me after.
No, just I was about to ask if anyone else would like to ask.
No, just in continuation to what he said, but
Yes.
in India, it is not possible, or it has not been done. So what are the main difficulties? It's
No,
easy for
no, I don't. I would not say that it cannot be done. I just think that like not anyone has picked it up right now. So I feel that like it requires you to set up It's absolutely no difficult at all. Like with I personally believe that we have got enough resources, enough talents, and particularly a country which is mostly focused on engineering and medicine. We don't have any dearth
So
of it. It's just that we have got
hardware-wise, what are the kind of costs involved? Hardware, what is the main thing you need? The
Yeah, so
software,
yeah, exactly. So this is exactly the reason I told that people who might be interested in entrepreneurship, this is an excellent area, because there is only one lab right now, because this Thor labs, or there are some other small labs coming up, but it has made such a monopoly market that whatever you want to buy components from there, like each of these optros or this optical fiber cables or the implants, they're quite costly. Like it's not something cheap like LED diodes, or though the principle is easy, but then it's not something very cheap. You would be spending quite thousands of dollars to set up like just one experiment, like, and they are fragile because you're putting on a moving mice, so they can break. So if it breaks, then you'll have to buy again from them. So like one experiment in which you are doing with some 30-40 mice can run into a couple of thousands of
this,
dollars, or like a few lakhs of, or more than few lakhs of rupees. So this is something which is a limitation right now, because like for me it's like like for going into developing countries like India, or every lab doing this kinds of experiments, like that's the limitation. But if you make the components cheaper, like the opto T cheaper, or the optical components cheaper, with mass production this is possible, or with more players coming out in the market it's possible. You can design your homegrown stuff. People do it sometimes because it's sometimes so expensive for Thor labs. People do it. We used to do it in my previous institute. We used to generate our own designed optos, but then again they are not quality controlled, right? So when you are like publishing a paper or something, people would be saying that like, was it quality control? So then you have to run a quality control experiment by yourself, which again will go into a lot of money. And so sometimes people feel it preferable to buy it from the companies. So I guess like that's the major limitation. But I feel that like institutes like in NCBS, my previous institute in India, CCMB, NBRC in Gurgaon, they are looking forward to this, and I feel that optogenetic setup should be coming down to India, and fiber photometry should be coming to India very soon.
okay, thank you.
No worries, you're most welcome.
Any other question from participants? Okay. In morning session itself, the first speaker mentioned that uh, Elon Musk is planning to work in this direction, and very soon means he will be in a position to download the thoughts of a human to maybe a
Yeah,
USB.
I would say that that is quite far-fetched. I don't see that's happening in the next 10, 15 years to download the thoughts. But yes, like the advancement of the field is going crazily. Like right now that there has been few labs in Japan and a few labs in the US where they put you inside an MRI machine, and they give you a setup, a present of images, like and then they try to get the map of your brain, an MRI or fMRI map of your brain, and then they try using machine learning to see what you have been thinking. So yes, like if it comes to like, it's preliminary, but yes, it's possible. So there are some neurons which is called the Jennifer Aniston neurons, actually. Like this was done because someone was presented a picture of Jennifer Aniston, and like those neurons started firing up, like when Jennifer Aniston's images were there, and that's the reason people started to recreate this. So like, yes, there are immense opportunities, possibilities. Machine learning is something which is going to help in all of these, and maybe in the future we will be able to do this, particularly in people who are coming from PTSD or any of these diseases, or if you are dreaming and you want to interpret your dream, you don't have to go to like trying to think. You can just go and sit inside the MRI machine, and you can just get your dream sorted out.
Yes, yes. So I think if there are no more questions, we can go ahead. So is the screen visible?
Yes, it's visible. Thank you so much.
So thank you very much, Dr.
Right.
Shiladitya, for
Thanks, Dr.
accepting
Amol,
our
for the
invitation and having a very nice session to all of us. So it was a new, rather a new field for all of us, rather many of us. And please accept this virtual memento from all
Oh,
of us. Yes,
right, sure.
from
Thank you so
SPIT
much.
and our team.
Thank you so much. Thank you.
Yes, yes, yes. Thank you very
Thank
much.
you, Shiladitya.
Thank you.
Thank you,
Okay,
sir.
so I'll sign off then. And you guys have a great session. I feel that it has been going on great. And
Yes,
hopefully,
yes.
like people will be inspired to come into research more often and have more research industry tie-ups than just,
Correct, correct.
like
So
science
that is
is
the main
different,
objective.
industry is different. Yes, and I feel that here in Germany they're doing a lot. So I hope India does the same too.
Correct.
Great. Thank you so much.
Thank you very much. Good day.